through. Okay, so this is a little bit about the whole blood, and so we have the blood cells are suspended in plasma. Plasma makes up about 70% of the blood. The difference between plasma and serum is that serum contains no clotting factors. And because they contain no clotting factors, they're really good for immune testing and immune therapy. So if you have anti-serum, for example, it's going to contain no clotting factors, which is good because you don't want it to clot in the needle or in your body once you get it there. You want the antibodies that are present to be able to do their job. So what you're seeing here is clotted whole blood, and so the area up above is serum, and there's some clotting that's occurred here, and so what's above this is the serum. So unclotted blood, you'll have the plasma and then a buffy coat and then your red blood cells down here, and basically this is what it looks like when you centrifuge it. But the plasma does have clotting factors, but this hasn't clotted yet. So here you can see some kind of clotting there that's occurred at the surface, and then above that is the serum. So the components of plasma, about 92% water, and protein is definitely necessary, including albumin and globulin proteins, general proteins, and antibodies. Also immunochemicals, fibrinogen, and other clotting factors. So fibrinogen is going to generate fibrinin, which makes the clotting occur. It sort of forms these network of fibers in the blood, and that allows it to clot. So we have hormones also, and of course nutrients, glucose, amino acids, fatty acids, any kind of nutrients that are necessary to be carried to the cells, ions that are necessary to be carried to the cells, and then of course dissolved gases, oxygen and CO2 are both in there, and waste products like urea, especially coming back from the kidney, I guess, or yeah, from going out from where things have started to digest protein. You're going to have urea before it goes to the kidney, because after the kidney it is in the urine. So anyway, waste products are also in there, like urea. So hematopoiesis, this is the production of blood cells, not just red blood cells, but all blood cells. And it begins in the embryonic development in the yolk sac. And so there's a little yolk sac that's in there in the 5th week. Very not the typical structure that you think about unless you've studied embryology. Normally we think of the babies as being like this, the fetus stage, but the embryo has this little embryonic yolk sac, and that's where it starts taking place. And then when it gets to the 8-week-old embryo, it begins to go to the liver. That's where you have it, because there are no bones here yet. And that is taken over by the liver and lymphoid organs at that point. Then they go into the red bone marrow. Now, notice the red bone marrow is in the long bones here, but when you get into the adult, it's in the ends of long bones. It's not in the, it's in the epiphysis, not the diaphysis, if you remember from your bones. So here it's in the shaft, and the long bones, the ends of the long bones are mostly cartilage at this point, not bone yet. And so when you look at a fetus, it looks like they're all disconnected because they're only showing you where the bone is. But the red bone marrow is in the long bones. It's in the flat bones like the ribs and the cranium bones, and then the pelvis, and then in the ends of long bones like your femur head is going to have a lot of this, and the others. And when they do a bone marrow transplant, then they're going to take it from an area that's easily accessible where there's lots of it. So they often take it from the ilium of the pelvis, the os coxa. So it's assumed by the red bone marrow in the adults, in the fetus and then the adult. But that is where the red bone marrow. It varies from the fetus to an adult and even an infant. So stem cells are the precursors. They're the beginning cells to the blood cells, and they're a pool of undifferentiated cells. Though they haven't decided which type of cell they're going to become at that point, and they're maintained in the bone marrow. And immature or unspecialized cells develop into specialized form and function, to develop the specialized form and function of the mature cells. So they start out these very non-specific. It's sort of like a freshman who comes in to college and they have no idea what they want to do. And so they take a couple of classes and they're like, wow, I really love biology, so maybe I'll be a biology major. And eventually then they get really specialized and they decide, I'm going to be a microbiology major. And so they get more differentiated and specialized to their particular function as they develop. Okay, so the development of the blood cells and platelets, there's this hematopoietic stem cell, and it can become this one type of stem cell that's made for all the blood cells except for the lymphoid stem cells. They become more differentiated, and you've got some that become, once they've decided they're an erythroblast, they become red blood cells. Once they have differentiated into this megakaryoblast, they become platelets. When they get to the myeloblast, they become these different kinds of white blood cells that are granular, and the monoblast, which becomes an agranular lymphocyte, or not lymphocyte, but an agranular, the monocyte. Then you have your lymphoblasts, which will make the B cells and T cells, and then from this lymphoid stem cell, a third natural killer cell is developing. So this gives you kind of an idea of what each of these different kinds of cells do and their relationship to each other, which is one of the things I like. It also differentiates between the granulocytes and what they do, but we'll talk about those more later. The mast cells are kind of, and every book I've seen this a little bit differently. Like some say that it's a specialized basophil, others say that it's a specific, like another, like a fourth kind of granulocyte, the mast cell. But it looks similar to basophils. It's got blue granules in it when it goes through the right stain method. And in any case, what it does is it's involved in allergies and the allergic response. So monocytes can become macrophages and dendritic cells, but in the blood they're called monocytes, and in the tissues they're called macrophages and dendritic cells when they go into the lymphoid tissues. There are specific kinds of macrophages, which we'll go over in another section. They become specialized in the various tissues, and they have special names for them. They're histiocytes, and they have different names for them depending on which tissue they specialize in. The lymphocytes then are the B cells and the T cells, and then of the T cells there's natural killer T and gamma delta T, which is a new one, and natural killer cells. These are ones that don't have any antigen specificity, which we'll talk about in the next chapter. So they're kind of non-specific because they are not antigen specific. They just kill anything that could be a virally infected cell or a cancerous cell, but they're not specific to a particular virus. They're not specific to a particular cancer. They're just anything that's, that's not supposed to be there, they will take it out. Natural killer T is sort of a hybrid cell. They have T cell antigen receptors, but they also have this natural killer, and so they call that kind of like a hybrid cell. And then there's this gamma delta, which is very new, responds to PAMPs, which are pathogen-associated molecular patterns, as well as specific antigens. And so there's a lot of these apparently in the gut mucosa. But these are something very new, so not something that they had in my book whenever we went through A&P, and so we'll talk more about that later.